video discusses the effects of the drug PTC 518, now renamed Votoplam, on Huntington's disease symptoms, and presents updated results from the ongoing Phase 2 clinical trial evaluating Votoplam, a splice modulator for HD. This drug is taken as a daily oral pill and works by modifying RNA splicing. RNA is the message our cells need to make proteins. Splicing the RNA is like editing the recipe before you cook. Your cells go through the message and cut out the parts, called introns, that are not needed, while sticking the useful parts, called exons, back together. Votoplam is designed to degrade the huntingtin RNA, leading to reduced production of the huntingtin protein. One of the notable advantages of Votoplam is its ease of delivery. As a pill, it provides an easy method of administration. It is able to reach both the brain and the rest of the body. Importantly, the drug is non-selective, meaning it lowers both the mutant, expanded, and the wild-type, normal, versions of the HTT protein. In this trial, Pivot HD, participants with HD at stage 2, showing clinical signs and symptoms, and stage 3, showing functional changes, were given either 5 mg or 10 mg doses of Votoplam, compared against a placebo group. The clinical trial had 2 predefined time points at which they would check the data, at 12 weeks and at 12 months of taking Votoplam. After the initial 12 months phase, all participants entered an open-label extension phase, during which those on placebo were switched to Votoplam, and all participants continued treatment for an additional 12 months, totaling 24 months of follow-up. Before initiating the trial, the researchers defined primary and secondary endpoints, which are measurable outcomes used to assess the effectiveness of the drug. The primary endpoint for Pivot HD was a reduction in huntingtin protein levels. Additional biomarkers included neurofilament light chain (NFL) levels, indicator of neuron death, and brain volume, which provide insights into brain health. At the 12-week and 12-month time points, Votoplam was found to be safe and well tolerated, with no serious adverse effects. Participants taking Votoplam demonstrated lower HTT protein levels, and the reduction was dose-dependent, meaning those that received higher doses had lower HTT protein. While data from the 24-month time point is still being analysed, early indications are promising. NFL levels, a critical biomarker for HD progression, was lower in participants who had been on Votoplam for 24 months. Specifically, NFL levels dropped by 9% in the 5 mg group and by 14% in the 10 mg group. This suggests that the drug may have a protective effect on neurons. Analyses using the Integrated Staging System, ISS, showed a slightly more positive effect in individuals with stage 2 HD. For those with stage 3 HD, the benefits were less clear, but no NFL spikes were observed, a sign that the drug does not appear to cause neuronal harm. Clinical outcomes, such as changes in the Composite Unified Huntington's Disease Rating Scale, CUHDRS, also showed encouraging trends. At 12 months, there were slight improvements in CUHDRS scores for ISS stage 2 patients. In stage 3, at 12 months, participants those on the lower 5 mg dose showed some improvement, whereas those on the 10 mg dose did not see the same benefit. However, at 24 months, dose-dependent improvements in CUHDRS were observed, suggesting that longer-term treatment may offer greater benefits. The HTT lowering ranged from 4% with the 5 mg dose to 39% with the 10 mg dose. The data imply that even modest lowering of huntingtin protein may be sufficient to achieve therapeutic effects. This is encouraging, since this is the first clinical trial that has met the primary endpoint. In summary, Votoplam appears to be a safe and promising treatment for Huntington's disease. While the drug lowers both normal and mutant HTT, it has not shown signs of toxicity or harm. The full impact on clinical outcomes and additional biomarker data, especially from the 24-month time point, are still pending. However, current results indicate a promising trend, particularly for individuals in earlier stages of the disease.